So my plan in the next 40 minutes or so is to go into the world of arbuscular mycorrhizal fungi to look what we know about their diversity and why it matters. Before I start really with the details of the diversity and fungi and all that stuff, I'd like to take a step back, and maybe you take a step back with me and look at this photograph. And look what you see. Think what you see, the obvious and the less obvious, and then maybe think what's behind it. So what is it that makes this possible? I see a family. I see a spot of nature, plenty of flowers. So obviously there is a soil which is quite okay. There must be water in the soil which is quite okay. There's probably clean air. There's probably a lot of biodiversity. I think these people have had food, maybe some porridge or some bread, maybe some vegetables such as carrots or tomatoes, maybe some coffee, maybe there was some chocolate. My mother might have had a glass of beer or wine the evening before, and they live somewhere and they have clothes on. So depending on what's your professional cretinism, you may think differently what's behind it. And I hope that by the end of my talk, you think a bit more with me that behind scenes like that, we need healthy soil and healthy soil biodiversity. Okay, so I want to talk about arbuscular mycorrhizal fungi, why they matter for plant growth. When I did my PhD, I started working with two beautiful plant species, and one of my questions was, are these plant species dependent on arbuscular mycorrhizal fungi? And here's a photograph of a pot experiment showing Pulsatilla patens, which is one of the pasque flower species, and Pulsatilla pratensis, another of the pasque flower species, growing without arbuscular mycorrhizal fungi. Okay? And you see a tiny plant here, this one. It has grown the same 3.5 months as these other ones, these plants and these plants. So what's the difference is that these tiny plants have been growing without arbuscular mycorrhizal fungi in the soil, in the roots. And the other ones have had different sources of inoculum of arbuscular mycorrhizal fungi. So it's very clear that these two plant species really require a symbiosis with the arbuscular mycorrhizal fungi, otherwise they simply do not grow. It's also very clear that these two different sources of the mycorrhizal fungal inoculum have a different effect on their growth. So these plants need mycorrhizal symbiosis, and they differentiate which fungi are in the roots. We know which fungi are in the roots because I sequenced them and identified them. So the fungi we're talking about are microscopic. You can't see them with the naked eye. When you look into the roots with some aids such as fluorescence, you may see fungal structures in the roots. Some of them are very finely branched, active and short-lived. We also see fungal structures around the roots, such as spores and mycelium. Again, these are microscopic. These fungi are present in most ecosystems, terrestrial ecosystems of the world. At the same time, there's not that many of them. So we know currently about 300 morphospecies, which are associating with most plants we know. As I said, these fungi are there in most terrestrial ecosystems, and they support the plants with the mineral nutrients. And in cases we find that all phosphorus, for example, a plant is taking up from the soil, has come via the hyphae of mycorrhizal fungi and not via the roots of the plant themselves. So these fungi are very important for the plant growth, for the plant nutrition, and it doesn't come for free. of course, these plants also use up some of the carbon. Sorry, the fungi use up some of the carbon provided by the plant to feed them. So the fungi are obligately dependent on the photosynthetic carbon provided by the plant. Which also means that both of these nutrient fluxes are important in the ecosystems. They are not minor. You can see the numbers are fairly large. It could be up to 1/5 of the entire plant photosynthetically fixed carbon that is spent on the mycorrhizal fungal partner. So it's a big proportion. So what I'm planning to do in my talk is to go through a couple of topics. I would start from really describing what we know about the biodiversity patterns of AM fungi in natural systems, in human influence systems. Then I go deeper into what our current agriculture does to these fungi and actually how could we better use them. And then of course we are left with the question, what next? So let's start with AM fungal biodiversity, what we know about it at the local scale, at the global scale, in the natural systems. So these are the two plant species I started working with as a PhD student. Very beautiful ones, also very long-lived ones. And our question was there, one of these species is very rare, and the other one is fairly common. They seem to go more or less in the same habitats. Our question was, is the very rare species very specific towards its fungal partners? So we made experiments, we sampled the field plants. We never dug up the entire plant because it's a rare plant, we don't want to kill it. And we identified AM fungi in the roots. And in these early days, this is my first international scientific paper, and it was published in New Phytologist. In these early days, we had small data sets, so we could show the entire phylogenetic tree of all the fungi we identified in our data set. And the things we were learning were very basic. We were learning which fungi were there. You see, there's not many names, there are some old codes. We could recognize whether they are related to known fungal species. We could recognize whether these are new, not previously detected, and how they are occurring in different habitats. So that was really the beginning of all these studies of molecular community ecology of vascular mycorrhizal fungi. So you see, I'm coding them, these fungi or fungal species, and I'm calling them phylogroups. At the end of my PhD time, I wanted to put my own data in a broader context. And again, as I said, the amount of data about the vascular mycorrhizal fungi globally was so small that essentially I could make a cluster analysis of all locations and plant species in locations globally. It was so few data. So each of the tips on this cluster analysis tree is a community of AM fungi colonizing one plant species in one location. And to my maybe surprise, maybe not, we found that these fungi seem to have underground biomes. So as we see forests and grasslands above ground, I could now see that the vascular mycorrhizal fungi from tropical forest are more similar to each other than a vascular mycorrhizal fungi from temperate forests. And a vascular mycorrhizal fungal communities, you can call them fungal vegetation or fungal biome from grasslands, are clearly different. So we learned that these fungi in the soil also have biomes. And in some ways it actually set the direction to many of the studies we have been doing after that. And one of the things we started doing was sampling globally. So we sampled natural habitats all over the globe to identify AM fungi in the roots of these locations. And you see that the world is not covered super well with these spots. At the same time, I think we started with these field works 2007, ended 2014. It took us years. We didn't have a dedicated project to that. So at that time it was a big effort and a big data set, and quite a lot of funds that went to sequencing the fungi. Okay, so we sampled the globe. And what we did with the samples from the globe, we sequenced the AM fungi in the roots over there and grouped them again into these sequence groups. And by that time, I had seen that I need to do something about that coding. So I'm walking you quickly through of our kind of species system that we use to be able to do community ecology of AM fungi. So these are real sequences, and I have run a simple phylogenetic analysis with these sequences, and you would see that they group into clades. You need to believe me, these are roughly species level clades, roughly. And some of these clades contain sequences from known species. So this is Glomus interradicis. Currently it might be called Rhizophagus interradicis. This is another one which again contains Glomus interradicis. This one currently would be called Rhizophagus irregularis. So we have here two clades on which we could put names and morphology behind it. There's another one here which doesn't have a sequence like that. So this is a DNA only DNA based clade. And you also see that individual authors who have found this fungus in their studies call this fungus very differently. Sometimes it's Globe 50, I've had it MOGE1, and so on and so on. So to get through this maze of coding, we generated the virtual taxon system. So now we are consistently talking about the same virtual taxon, which is about species, to be able to do community ecology of AM fungi. And the system is available in a public database called Names After Me, with publications explaining the principles of it. So back to the communities of AM fungi all over the world. With this global sampling, we learned a lot, and some of the things we learned was that many arbuscular mycorrhizal fungi are widespread. And by the widespread, we mean that the same virtual taxon or species can be found on multiple continents. So we found that over 90% of the virtual taxa were present on more than one continent, and 1/3 of all the species of virtual taxa were present on all the continents we surveyed. So 1/3 of the species have a global distribution. This doesn't mean that everything is everywhere. There are regional differences, and I'm getting to this in the next slide. So here we are looking at two patterns, and one of them is the Richness pattern across latitude. On the left side, you see absolute latitude from the equator to the north or south higher latitudes, and then the richness of AM fungi in the plots over there. And you would see that both in the forest, which are the red symbols, and the grassland locations, we see that the tropical latitudes harbor higher diversity of AM fungi than the temperate latitudes. So we discovered latitudinal diversity gradients for these organisms as well, as known for plants, for example. But also we discovered that the habitats differ. The forest and grassland habitats seem to differ in something very fundamental. The right side graph is showing the community composition of the forest and grassland habitats, and then what would be the main predictors of this composition variation. And we really see that weather variables such as precipitation and soil variables such as pH or organic carbon content are influencing the communities of AM fungi quite a lot. Okay, then we of course asked, why are all these fungi on all continents, or some of them? Is it because this is an ancient group of fungi? Have they simply had a lot of time to disperse. As I said, there's quite a small number of these fungi, about 300, or we had less than 300 in our data set. So maybe they just had a lot of geological time to reach the continents. So here we ran a phylogenetic tree. This is a single gene tree, and it has only a single calibration point at the root. My conclusions are in Estonia. I hope you enjoy them. And with this phylogenetic tree, we were asking whether most of the tips we are seeing currently, meaning most of the species, did they evolve before or after the last continental refiguration, which is indicated by this pale blue line over there. And you would see that most of the tips have evolved after that, meaning that the continents as we see them separated now were there, and the species which evolved and are now found on all continents must have dispersed to these continents, meaning that it's the dispersal and not ancient speciation which is explaining the current biodiversity patterns of AM fungi globally. Let's jump back into local diversity patterns. And here is a study run by one of my PhD students, Sepp, and he went to this grassland in West Estonia, which is very species rich. It looks a bit monochrome. Actually, it could be very colorful now and then. It's a dry calcareous grassland, which is a typical habitat in Western Estonia. And he sampled all vascular plant species in that habitat 2 times in a year, and sequenced AM fungi from the roots of these plant species. And he was interested to learn whether the plant species in some regular way interact differently with the AM fungi in the roots. And you see here each of the bars is one plant species, and on the y-axis we have the mean number of AM fungi per sample in the roots of these plants. First of the things you see that there's a lot of variation. These plants are in the same grassland patch. They don't have the same exact number of AM fungi in the roots. They also don't have the same exact set of AM fungi in the roots. So they do differ. But is there any clear kind of pattern how they differ? Here we have colored the plant life forms. So the green ones are forbs, blue ones are grasses, and then we have one fern and 2 shrubs over there. We tested many different predictors, and quite annoyingly, it was not super clear. One of the differences we detected was the difference in AM fungal interactions between forbs and grasses. So you see that the grasses seem to harbor on average more AM fungi than the forbs. I would like to stress that this might be specific to this data set. Not sure that this would hold up if we would expand it. At the same time, we had quite a lot of samples. There's a good replication. So clearly in this study site, grasses have more AM fungal species associating with them than the forbs. We also tested whether the plant abundance matters. So on this bottom graph, we see the local plant frequency and how this relates to the AM fungal diversity. And we see it relates positively. So the more frequent the plant species is on this grassland, the higher the richness of AM fungi it tends to have in its roots. So we can see that the plants and AM fungi do associate non-randomly. There are some regularities about it. Plant life form might be one of the predictors, and the plant abundance might be another of the predictors. What this exactly means, of course, may require further study. And this may also depend on the context, how the plants and the AM fungi interact. And for this context, I would like to make a little step into concept of species pool. So I'm showing here a map of AM fungal diversity globally. So the warmer colors indicate higher diversity and cooler colors indicate smaller diversity. And we measure it in the terms of species pool size. What is it? The concept of species pool was proposed in 1990s by Martin Sobel, who is the head of our team, but also independently by some other teams. And in essence, it's trying to explain how the local number of species is explained by, first, how many species have evolved, how they disperse, and how they can reach the local habitat. So in other words, to which degree the local number of species depends on the regional and historical processes, be them evolutionary or biogeographic. And the species pool concept could be visualized like that. We would have species in the local site. We would have species in the neighborhood. They build up in time. And we observe a set of species in the local site, which is a subset of species available in the neighborhood. Important aspect of this is that the local site has its own conditions. So not all species in the neighborhood would actually fit with the conditions of the local site. So this means that there is a set of habitat-specific species that could inhabit the local site at any given time. And always some of them would be present and some of them would be missing. And therefore we are recognizing these components of the species pool, which is the observed diversity and the dark diversity. And if you may, if you think about these terms in terms of humans or maybe the attendance of this conference, then the species pool of this conference would be all of the people who have registered for this meeting. The observed diversity are all of you who are here in this room now. Some of you are not here. Some chairs are empty. So these people who are currently not here, but they have registered for the meeting, they are the dark diversity. They could be here, they have the capacity to be here, but in various reasons they are not. So that's the concept we are using for describing the diversity of fungi or plants. And sometimes it's quite useful to make a difference between these two sets, the observed and the dark diversity and the total species pool. So one of the conditions when it's useful to make this difference is to see how the species in the region have adapted to a specific habitat. So for example, AM fungi globally relate to soil pH differently depending on whether they are from temperate or from tropical regions. And what this means is that in the tropical regions there has been more time for species to evolve who adapt to all kinds of pHs, whilst in the temperate regions the species of AM have more adapted to the higher pHs, but less so in the lower pHs. And therefore we see a smaller number of species at the lower pH. And therefore we also can see how the number of species globally in the species pool and the number of species in a location may be driven by different factors. So here we look at the same AM fungal species pool. I showed the map of you before, and we see that the largest predictors for whether we have a big species pool in, say, Brazil, or a smaller species pool in, say, North America, is a lot determined by the biomes that we had in the neighborhood during the last glacial maximum, meaning the historical factors. Whilst the number of species we observe currently are determined a lot by the current processes. So here we are looking at the observed diversity, and this is influenced by which biomes are in the neighborhood now and how wild or anthropogenically influenced they are now. So the local diversity as we observe it now is determined by current predictors, and the potential diversity, the species pool, is influenced by historical predictors. This may matter when we think about anthropogenic systems. So here we look that everything I've been telling to you up to now has been about natural systems. Here we started looking at what happens when we turn a natural system into anthropogenic one. And again, we made global sampling, and we sampled forest habitats and grassland habitats. Here they are woody and unwoody. And then in a paired manner, next to each of the forest or grassland habitat, we sampled something that was anthropogenically influenced. For example, arable fields. It could have been a golf course, it could have been something else like that. And then what did we find? At first we were again a bit perplexed. It looks like the anthropogenic influence is very small and doesn't really do anything to AM fungal diversity. So again we learned that the grassy or grassland habitats had a higher diversity of AM fungi, so it's again the mean richness in the sample, and the forested or woody sites had it lower. Okay. But then we started comparing in each of the cases the natural and influenced site. And we see the result of this comparison here. So on the x-axis we have the average richness of AM fungi in the natural plot in this region. And in the y-axis you see the change. So again, the brown colored sites would be the wooded sites. And in the case of these sites, you would see that turning the habitats into, for example, arable field tends to increase the AM fungal diversity. Whilst in the case of the very rich AM fungal habitats, the grasslands, if we turn them into arable fields or other anthropogenic sites, they lose AM fungi. They lose the richness. So hence, in a way, we are applying a McDonaldization to these communities of AM fungi globally. The context is important. The forests and grasslands behave very differently all over the world. Also in the regions. So here's a study comparing the forests and grasslands and arable fields in Estonia. And you could again see that grasslands are more rich, forests are less rich, and arable fields, they depend. So sometimes they are less or more diverse. This less or more diverse may also depend on the history of the land use. And here if you look at the bottom graph, it's looking at the AM fungal diversity in the dry grasslands, the same alba grasslands in the western Estonia. And for these ones, we know super well how big the areas of these habitats were earlier. So we could map the current richness of AM fungi in a site depending on how big the area of the habitat was historically. And you see these top red and blue lines, which are from open and slightly overgrown alba grasslands. They have higher richness if historically the habitat has been larger. We can turn it other way around and say they have lower richness if the area of their habitat has been historically smaller. And currently, of course, the habitat is decreasing and decreasing. So you just draw the line and you see that there is a danger of extinction of many of these fungal species in the soil because we are losing their habitat, the grasslands. So both the soil fungi and plants, as shown earlier, they do exhibit extinction depth. Quickly about what does our current land use do to AM fungi. So here I'm presenting you a study from a European consortium funded by Biodiversa, and this set of labs is looking at the field sites, arable field sites, and the biota in the soil in function of how the tillage is applied. So we use the usual tillage, a very intensive tillage, all the intensive management, and then there are sites with reduced tillage, which are hopefully going to be better for the soil biota, such as earthworms, fungi, AM fungi, bacteria, and all others. and we are looking how it really is. So just briefly, the top 5 AM fungi we find in these soils, in the fields, experimental farms across all these countries. For me, this list shows 2 things. One is that 2 of the 5 are the fungi which we don't know morphology of. So we have here this DNA-based Glomus and the DNA-based Diversispora. Another one is that there's one fungal isolate of species, Rhizophagus irregularis, which is very highly used in experimentation. It's a model fungus of the group. It's not there. It's not there. It's not common in these fields. So essentially what we learn from this model fungus could, and maybe it doesn't apply for the fungi we see commonly in our field sites. But back to the tillage. So we're looking here, did the reduced tillage, which should be better, it's not disturbing AM fungi that much. Is it improving the AM fungal diversity? So here is the summary of the AM fungal richness, mean richness per sample across all countries in the conventionally tilled sites and in the reduced tillage sites. It's very obvious there's no change. But when we plot the community composition of AM fungi across all these countries, then we see that the countries differ. So each of these colors is representing a country. So you see that individual countries have more influence, meaning regional set of species available for these farms. There's more influence on which fungi are there than the actual management approach at the European scale. If we go and look into the individual countries, we see a lot of diversity in terms of the directions of the effects, and I'm just showing you 2 of them. One is France, where we see a reduction of the diversity of AM fungi when we reduce the tillage. Why should it be? I get to this in a moment. Another one is Sweden, where we see that there is a trend for increase in AM fungal diversity if we plow them less, if we disturb them less. This is somewhat expected. Of course, we started thinking, okay, but then what is the system size? How many species we have in these systems? And this plot is looking across the number of samples, how many species of AM fungi we observe, and we see that the countries do differ. So some countries have more species available, others have less in these farm sites. So it's not to be concluded about the countries as whole. And this may mean that if we change the tillage, then these dark red-colored AM fungi are the ones which we are going to lose if we change the tillage to reduced tillage, and the yellowish ones are the species we are going to gain. So here in the case of France, by reducing the tillage, we lost more species than we gained. And maybe these species that we could gain in the reduced tillage system are not there any longer. So we need to think of the context, so it's the species pool, but also the historical management in the region when we think about what is happening with AM fungal diversity in the sites. And this is what we can also plot at the global scale, comparing the species pools of natural habitats. Here is a Europe and Asia comparison. Commonly, the observed richness is about 20 species per site, whilst the size of the species pool for natural habitats tends to be 2 times larger, both in Europe and Asia, in natural habitats than human-influenced habitats. And the total overlap of the species of AM fungi that can grow in both natural and anthropogenic habitats is small. So there are different AM fungi which can grow in these habitats. So again, we really need to understand better the context of which fungi can grow where, under which conditions. And then we can think how we can use this fungi better or manage this fungi better. And one of the examples is a restoration study in Estonia in semi-natural grasslands, which tend to overgrow if we don't use them for grazing, for example. And here's an experiment by another of my PhD students, Tanel Vahter, where we are using game fungal inoculation to restore semi-natural grasslands and abandoned quarry sites. So the system is such that we use locally produced very crude game fungal inoculum, or the same thing with plant seeds, or only plant seeds, or nothing at all. Here we use mulch as well to just help the added inoculum to stay on the sites. And you can see that these are moonscapes. So these birch trees on the back, on the habitats, they've been planted there to reforest them maybe 20 years ago. They are super unhappy, these plants. And we have followed these sites now for 2 years, and one of the things we see is that we managed to inoculate AM fungi there. So in the inoculated sites, we observe increase of the richness of AM fungi by about 2 times. And the same is with the plants. So the plant diversity also has increased by about 2 times, which is a very good news. So it shows that there's a promise that this kind of a practice can actually help to restore the vegetation in these abandoned quarries, and the same with grasslands. So here's a vegetation we have managed to, well, which grows on the inoculated plots, and it's quite high, quite big, fairly diverse. And you see this moonscape behind the plots. This is the second summer. I would say it's a striking result. We keep monitoring. Okay, where next? Human health is influenced by soil biota health, and we can do different things to maintain or damage the soil biota health. We can also see how it matters. I'm quickly running through a study which is looking how forest biomass, meaning the amount of timber, is related to local soil biota. And it was really big and intensive study in South Estonia in 100 forest sites. And my colleagues were sampling plants, bryophytes, lichens, soil, from which we sequenced fungi and AM fungi, all fungi and AM fungi separately. And then some of these forest sites are very wild looking. And then looked how this diversity of organisms relates to each other and relates to the timber mass. And here you see that timber mass or forest wood volume is mostly explained, or maybe you don't see, mostly explained by biodiversity. There's a lot of unexplained variation, but about 10% of the variation is explained by biodiversity, and all these other things like soil and climate don't really explain the timber. So it's a message to the forest growers or forest owners that biodiversity supports the amount of timber they can get from the forest, which is important. And also the relationships among these organism groups over there, different groups of fungi. We had pathogens, ectomycorrhizal fungi, avascular mycorrhizal fungi, and so on and so on. They do relate to each other. So there's a lot of complex interactions between diversities of organism groups in these common forests, temperate forests in Estonia. That's all I wanted to say, really. I hope I'm roughly in time for you to ask some questions. But so coming back to what I started, where I started from, I say that if we want to have landscapes which make it possible for us to live on this earth for a little longer and have some food and beautiful clothes and nice homes and coffee and wine or carrots, then we need healthy soil biota. Thank you.